All right, thanks everyone. Thanks for coming on this gloriously snowy early November day. So I'm very excited to present Liz Herrera, who's a PhD candidate in my lab. Liz has done really great work tying together a really cool model of stress susceptibility, really cool methods in terms of analysis of structural and fMRI data, and on top of that, really cool results on sex differences in stress susceptibility. So it's like it's a bit of a banger of a thesis. This is one chapter of her thesis that she's going to present today. It's a biggie. So thanks for coming. And Liz will likely be, well, we're just discussing this now. She will likely be defending either the end of this year or probably next year. So any comments you have, any feedback you have, this is a great time to give it. This is a great preparation, I think, thing for her to be able to do and have all this brain power in one room to help her out. So, be as friendly but as critical as possible. And I think Liz will greatly benefit. So thanks you all. Thanks to all of you for coming today. All right.
Thank you. Okay, so thank you all for being here today. So today I'll be discussing my work, which is entitled Dissecting the Role of Sex and Stress Duration in Individual Responses to Chronic Variable Stress. So as many of you may be aware of, depressive disorders are characterized by the presence of depressive mood or loss of interest for at least 2 weeks, in addition to 5 other symptoms which are found here on this list. And one of the things that you can notice in here is that first, there are many combinations of symptoms that patients can experience, meaning that not every patient with depression will experience depression in the same way. And in fact, people have found more than 100 different combinations of symptoms across patients. And importantly, differences among depressive patients are found in between sexes. So we can see that women are almost twice as likely to be diagnosed with depression. We also have differences in symptom profiles, in comorbidities, but most importantly, in the way they respond to one of the major risk factors for depression, which is stress. And when we look at the number of publications per year, we're using neuroimaging and looking at depression, we see that there's been this increase over time in people trying to find biomarkers of depression. However, given the marked sex differences that we see in this disorder, there are not a lot of publications including sex as a biological variable and looking at sex differences or sex-specific mechanisms. On the other hand, when we think about stress, this major risk factor for depression, there are many ways that a person can experience stress and respond to it. And there have been many theories proposed about how adaptations occur upon a stress exposure. And we can think of this as a linear relationship, where upon increased stress exposure, there is increased risk for depression. On the other hand, we can think of this as a threshold type of thing, where upon the first experiencing a stressor, this is no longer related to an increased risk for depression. Or we can think of this as a trigger that then increases the vulnerability of that individual to future stress. So the first stressor was bad, which seems like really bad, and the next ones are not that as bad. It still increased the risk for depression. But only in that, these different responses that people have proposed vary across individuals. And this can vary depending on the genetics, biological sex, and also the environment that the person is found in. But my thesis mainly focuses on one of these factors, which is biological sex. And in order to study this, we use mouse models for depression. So in this case, I'm focusing on the model known as chronic variable stress, in which this model, the goal is to mimic the human condition when we are faced with inescapable stressors. So mice are exposed to food shocks in one day, tail suspension on the next day, wrist strain on the next day, and we repeat those stressors throughout the course of days to weeks, and we see how there are different adaptations, either in behavior or in the brain. But one of the important factors that decided that we used to decide whether using this model or other models of depression was that this model has revealed that this very dynamic interaction between duration of stress exposure and biological sex, in which specifically we see that if we expose male and female mice to different durations of stress, we can induce similar behavioral phenotypes, such as in anxiety-like behavior or in social preference. And we can also induce similar neurobiological mechanisms in regions, for in this case, the ventral hippocampus. On the other hand, if we expose both males and females to similar durations of stress, interestingly, we see now sex differences, even though they were exposed to a similar paradigm, either in regions that are like the nucleus accumbens, the ventral hippocampus, or the ventral tegmental area, suggesting that we have this jointly relationship between sex, stress duration that are shaping vulnerability to stress. Also, I forgot to mention that you can make any questions during the talk. However, most of this work has been focused on doing just case control analysis, where you have a control sample, a stress sample, and you just compare them. Other people have looked at this into like a resilient and susceptible phenotypes and just making these two binary distinctions between stress mice. However, when we think about this, is that stress responses are really variable across mice, and we can think as from a dimensional perspective, where each mouse deviates from the control population in a very different way, and not just in one axis, but across multiple axes of deviations. And importantly, one of them is biological sex that makes them fluctuating their stress responses. So the goal of this study is to examine sex's and dose-dependent responses to stress in behavior, physiology, brain structure, and function. So we do that by exposing 3 different cohorts of male and female mice, either to 6 days of stress, 15, or 28 days. And we first acquire behavioral tests of anxiety-like behavior at baseline and also after stress using the open field test. We use the social preference test to assess social withdrawal. And we also acquire structural MRI before and after stress, and functional MRI after stress, in addition to physiological metrics of heart rate and respiration rate. So we took the classic approach first for the behaviors. When looking at the open field test, we look at time spent in different zones. When we're looking at the social preference test, we look at how many times they interact with a novel mouse or just an object. But we wanted to take it one step forward because these measures can overlook naturalistic behaviors. So we use a data-driven behavioral approach using deep lab cut and key-point mosaic to define naturalistic behaviors that are happening when the mouse is in the open field. So by using deep lab cut with help of the undergrad London that did all of this work, we tracked the body parts of each of these different mice, the nose, the shoulders, the tail. And then after tracking, we use key-point mosaic to derive 40 behavioral syllables that are characterizing different behaviors that the mouse is doing during the test. But this is like a lot of behaviors that are happening. It's really hard for a human to really grasp what are these mice doing during each of these behavioral syllables. So to reduce this high dimensionality of the data set, we did an exploratory factor analysis, and we decided to keep 3 factors that were describing most of the variance, and we examined how they were changing upon a stress exposure. So the interesting here, first is that we see that there is increased anxiety-like behavior if we look at classic measures of anxiety-like behavior in the open field. So there is increased anxiety-like behavior across 6, 15, and 28 days of stress. That is similar both in males and in females. So here you're just looking how they differ from their, within sex group differences, so how they're different from their controls, either at baseline or after the different durations of stress. But the important thing is that despite seeing here the similar phenotype in both males and in females, when we look at the results from the factor analysis from the behavioral syllables, we see different things. So here I'm showing you the different syllables, how they're loading onto different factors. And here's one example of those behavioral syllables that are highly loaded onto this factor. So when you see the white dot over there, is like when the behavioral syllable is happening. So these are different behaviors that the mice are doing during the open field, but we are not able to capture by the human eyesight. And interestingly, we do see sex differences in these factors. So these, we decided to call it head scanning behaviors in the borders. So the mouse is doing a lot of head scanning, specifically in the borders of the open field arena. And we see that these behaviors are increased in males only exposed to 6 days of stress. Yeah.
About this to make sure I'm understanding it. Okay, so the factor analysis, essentially what it's saying is that there's like a lot of variability in whether the mice are doing this, like whether they're doing it or not, this behavior of like scanning in the corners.
No, the factor analysis is mainly to group all these behavioral syllables into something that is more interpretable. So all of those syllables that have a high loading in here, they all look similar. So they're all doing this type of head scanning behaviors in the borders of the open field. So it's just mainly to group them, like cluster them into something that is more digestible.
What does it mean that this factor is one of the three that you were looking at? Like what? Why choose three?
Well, mainly the reason that we chose three is because of the scree plot. That's like the main reason.
Sorry, what?
The scree plot, like the elbow
Okay.
curve, but yeah, that's like the main reason that we only have three factors.
So
Yeah.
like those three factors like explain more of the.
Yeah.
Is there something about explaining variance in the factor analysis?
I think there is. Yeah, I didn't include it here, but yeah, I think there is like about
Okay.
explain marks. Yeah.
And what about like the because you have both behavior and then position in the open field, right?
Yeah.
So like what is there also like this behavior but not in the border?
Yeah.
Okay.
Yeah. So the same behavior, but being done in the center of the open field. Yeah.
Okay. Thanks.
And that's actually like the next factor. So we are just looking mainly at behavioral syllables when they're being done in the center of the open field. And now we see that both males and females are doing less of these crouch and scanning behaviors in the center following 6 and 15 days of stress. And for the last factor, which was similar to factor 2, but now this is similar behaviors, but now that are being done in the borders, we don't see any differences upon a stress exposure. But then we wanted to link how these behavioral syllables are associated with the traditional metrics that we have of behavior, of anxiety-like behavior or social preference. And for that, we decided to use mean center partial least squares. So just like a brief summary of how this works is that you have your set of behaviors across all subjects. Then we calculated group averages. So for each of the durations, sexes, or groups. And then these were subjected to singular value decomposition that yielded a set of latent variables, each characterized by a contrast pattern that tell us which group is loading significantly to this latent variable, and also a behavioral pattern that's telling us which behaviors are loading significantly onto this latent variable. So we identified two significant latent variables. So here I'm showing you the first one. So these are the squares that are in gray are not significantly associated with this latent variable. So this is a latent variable that is mainly characterized by locomotion during the open field and also head scanning behaviors in the borders of the arena. And if we look at the group loadings in here, so here you're looking on the top for controls, on the bottom for stress mice, and in the different types of points are for the different durations of stress. And those that are shaded are not making significant contributions to the latent variable. So in here, what you can notice first is that this is a latent variable that is significantly expressed only by females following 6 and 15 days of exposure. Then for latent variable 2, it's a latent variable that is mainly characterized by increased anxiety-like behavior measured by time spent in the center, also changes in their social preference, and also crouch and scanning behaviors in the center. And importantly, we see now that it's a latent variable significantly expressed by only males following 15 and 28 days of exposure. And overall, what this analysis is showing us is that we are maybe capturing behavioral susceptibility that has been suggested previously in the literature to emerge early in females, late in males, in which we can see that it emerges first at 6 and 15 in females, and then at 15 and 28 days in males, which supports previous findings. But this is one way to look at behavioral at stress susceptibility. And actually, there are a lot of things happening in the body. So if we first look at the physiological adaptations that these mice have upon a stress exposure, we see that both males and females are losing weight. So they're gaining less weight compared to their controls. And this is observed across all stress durations. We also see differences in heart rate variability. So we see that females exposed to 28 days of stress have lower heart rate variability, which is a marker of their autonomic system is less adaptive to stress. And lastly, we see differences in mean respiratory rate in males at 15 days and females at 28 days. So meaning that this is, even though like we saw this behavioral susceptibility emerge early in females, late in males, this is not parallel to what we saw. These findings are not parallel to what we saw in behavior. So it reflects that maybe likely in the brain, there are other signatures happening that may be driving these differences, this dissociation between behavior and autonomic responses. And for that, for brain structure, so I'm going to switch gears a little bit, we decided to take an approach that has been recently been done in humans. So I don't know if you've heard about normative models. So normative models are a way that we can do kind of like brain charts. So if you go to a pediatrician, the pediatrician will measure your child weighted and then tell you how far it is from the mean population, how many standard deviation your child is away from what is expected to be at that age. So we can do these brain charts for either different tissues of the brain, gray matter, white matter. But this has been only done in humans. And we decided to do it for the first time in mice. So by using all the data that we have from all these 3 different cohorts, we can measure their brain volume and then create normative models of brain volume. And specifically, we can do it sex-specific. So we can do a model for males, a model for females, and then map our stressed animals onto these normative curves and derive individual level maps of volumetric deviations that were induced by a stress exposure. So the way that we did this is that we use, to build the normative models, is that we use deformation-based morphometry. So we use the scans from only the control mice across the different ages that were collected. We created time point-specific templates using these control scans, and all of these were mapped onto a common space template. And then we can use the deformations that were used to create these time point-specific templates to derive either volume increases where a voxel had to shrink or expand. In this case, if the voxel had to shrink to fit the time point-specific template, we estimate it as a higher volume, whereas if the voxel had to expand relative to the time point-specific template, we are defining this as a lower volume in that voxel. So this is how the normative models look in this, either for males or for females. So this is just one voxel in this case for the infralimbic area. So as you can see in here, so I don't know if you can see it. Oh, sorry, the projector was too small. So, so the gray dots are the control mice. So these are the normative trajectories for males and females. And here, either the pink dots or the green dots are reflecting the stress mice. And you can see how mice are deviating differently, in this case, in the infralimbic cortex following a stress exposure. So these are for the different durations of stress where we can find these mice. And one thing that we noticed first when we were looking at this data was that there was a lot of variability in how these mice how the brains of these mice were adapting to stress. So here you're looking at the percentage of subjects that have deviations that are greater than a Z-score of 1.5. So here at the top, you see either positive deviations here at the bottom or negative deviations here at the top for both males and females. And one thing that we noticed was that as we were increasing stress exposure, mice were converging onto a set of deviations that were found in the cortex and in the thalamus. But even though they were converging onto these deviations, there was still a lot of variability between mice in how their brains were adapting. And in fact, when we do just a group level analysis, we do find that this increased spreading of volumetric deviations across the brain that initially starts upon just 6 days of exposure in the hypothalamus, a very small dot that you cannot see there, but it was just one small dot in the hypothalamus, and also in the lateral septum. Then upon increased stress exposure, we start to see that it spreads to more cortical limbic regions, hypothalamic regions, and it spreads more following 28 days of stress. But if you take these 2 findings, these group level findings, and the other one from the individual variability, it means that these mice, either like, even though there seems to be converging onto a specific circuit that is being altered by stress, there are many ways that we can get to it. And for that, we use, again, mean-centered PLS to find out how these different mice are getting onto this convergence of deviations. So the first Latent variable from this analysis show that we have this first spreading that we see these dose-dependent alterations that are observed. So these are the brain scores. So each dot is a mouse in here. So I just want to show you this because for you to see how variable these mice are, even though they were exposed to the same paradigm and even though they're supposed to be genetically identical. So what you can see here is that first, males and females that were exposed to 15 and 28 days of stress, they differ from the controls in the way that they express this latent variable of increased volumetric deviations in these regions. But if you look at the next latent variables, for instance, so this is a female-specific latent variable that is affecting subcortical regions, including the lateral septum, the dorsal hippocampus or thalamic nuclei. We see that their female mice begin to separate from controls at 15 days, but not at 28 days. Then for latent variable number 3, this was a latent variable, but was characterized by separating males and females at different time points of stress. So we can see here that males separate from the controls after 6 and 15, whereas females separate from the controls at 15 and 28 days, which may be paralleling the findings that we had from our behavioral analysis that have these different time points of alterations. And then lastly, for the last latent variable, this was a male-specific latent variable, which was specifically observed after 15 and 28 days of a stress exposure that encompass prefrontal regions and also regions like the VTA. Yeah.
What do you think the changes in the directions? So, from like 15 to 28, for example.
I don't know.
It's challenging.
Yeah. Yeah. So
is one priming for the other kind of thing? Is that like one way to think about it?
Could be, or could be like
Holding you back.
yeah, like something that just gave up. It could be. So in order to understand what these different latent variables may be reflecting in terms of biological processes, we decided to use a spatial gene expression analysis using the Allen Brain Institute dataset. So we look at the spatial correlation between each of these brain patterns and spatial gene expression patterns. And then we rank these correlations, and using these rank correlations, we perform gene ontology at the end, at genes that are at the extreme ends of the list, when we compare them against a null distribution that we create from random phenotypes. So we create a random distribution by just, a random phenotype is the average of random genes. And then we, for each of these random phenotypes, we calculate the Spearman correlation, repeat the process of ranking them, and then we come up with this null distribution for each of the gene ontology terms that we can define whether or not the gene ontology terms were significantly associated with that brain pattern. And what we find in here is that for each of these latent variables, we have different biological processes. So for the first one, we find that it is associated with these two gene ontology terms that we think that it might be related either to inflammation, meaning that there is higher recruitment or migration of immune cells that then change their shape and become either activated, reactive, or it could be also just neuronal remodeling that is happening in these regions. Then for latent variable number 2, we observe that it is associated with terms associated with vasoconstriction, mainly heart rate, smooth muscle, also insulin receptors, response to ketones. So maybe the females at 15 days of exposure, maybe having this autonomic regulation mechanism that may be giving up after 28 days of stress, where we see these differences, for instance, in heart rate variability. Then for the next one, it was associated with pituitary gland development. So meaning that it is not parallel to our behavioral findings. It's actually going the opposite way as our behavioral findings. It might be suggesting that these males and females have different responses in the HPA axis that are driving how their behaviors are either emerging or disappearing. And lastly, for the male-specific Latin variable, we didn't identify any significant associations. But what these findings are suggesting is that we have this group-level dose-dependent response in neuroanatomical alterations. But the way that each mouse gets into that dose-dependent response is subject-specific, and that is modulated either by duration of exposure or by sex, and that each of these different ways that they are getting into that reflects different biological mechanisms. So now if we look into our functional MRI, so now we had functional MRI that was only acquired at the post-stress time points that was pre-processed using the in-house pipeline, rabies. And then we use, to derive resting state brain networks, we use a technique known as probabilistic functional modes, in which essentially the way that it works is that it decomposes an fMRI time series into a set of, for each subject, into a set of spatial modes, mode amplitudes, and mode time courses, which reflect the spatial distribution of each network, the relative activity of that network, and also how that network was the activity of that network across time. So we found 6 different resting state brain networks. This included the somatomotor, somatosensory, salience, one part of the default mode network, another part of the default mode network, and the limbic network. And we decided to look at how these brain networks are getting reorganized following stress. And for that, we look at the spatial correlation just between all these different set of networks. So in this case, I'm showing you an example of what the spatial correlation would look like for 2 different subjects between the somatomotor and the default mode network. And you can see here that for one subject, these 2 networks become more together, I'd say. Whereas in other mice, these networks are more segregated. So meaning that their spatial correlation is lower. And similarly to our previous analysis, we use mean center PLS to find these patterns of how brain networks are getting reorganized following stress exposure. In the first Latin variable, what we find is that the Latin variable significantly expressed by males and females following 6 days of stress. And it is characterized by increased spatial correlation between sensory and association networks. So for instance, between the somatosensory and the salience, the DMN, and the limbic network. On the other hand, Latin variable 2 was significantly expressed only by males and females exposed to 28 days of stress, but in a sex-specific way. So males express the opposite pattern that is shown in here. So instead of having increased spatial correlation between association networks, as it is in females, males are having more segregation between association networks. And now that I've shown you like how this behavior, physiology, brain structure, brain function change altogether upon stress, we wanted to ask the question, well, how are all these things related? And for that, we use partial least squares correlation. So in this case, what we do is we're looking at patterns of maximal covariance between 2 sets of variables. So in this case, we're looking at brain-derived measures, either volumetric deviations derived from the normative models, or the spatial correlation between networks, and all the behavioral and physiological metrics that we have. So we first look at compute the correlation between these 2 variables, and then we submit this into a singular value decomposition to obtain a set of Latin variables, each characterized by a behavioral pattern that tells us which behavioral variables are significantly loaded onto this Latin variable, and a brain pattern that tells us which voxels or brain networks are significantly associated with this Latin variable. So if we first look at structure-behavior relationships, so this is the behavioral pattern that we find in the significant Latin variable, which is mainly, so these are the different behaviors. Each is color-coded depending on what it might reflect to, either anxiety-like behavior, social preference, the type of behavioral syllable, or physiological metrics. So we see that it is characterized by increased anxiety-like behavior, changes in social preference, and increased expression of crouch and scanning behaviors in the center, which were associated with volumetric changes in cortical, thalamic regions, hypothalamic regions, and was specifically associated in stress mice. So we see a dose-dependent expression of this latent variable in both males and females as we increase the stress duration. So as you can see in here, these are the brain scores or the behavior scores for each mouse, which reflect how much that mouse is expressing this latent variable. And we can see that as we increase the stress duration, these mice express more and more this pattern. So we have more anxiety-like behavior and also more volumetric deviations, for instance, in both males and in females. But then if we look at the relationship between function and behavior, we see a latent variable mainly characterized by just the stress exposure. So the first latent variable was significantly observed only in females. And this one was characterized by changes in locomotion, both during the social preference test and open field, crouch scanning behaviors, and some physiological metrics like mean heart rate variability, or respiration rate variability, and was associated with increased spatial correlation between sensory and association networks. We can see, and when we look at the brain and behavior scores, we only see that there was a significant association in control females, meaning that stress exposure disrupts this association from what is expected to be there, but regardless of the duration of stress. Then for latent variable number 2, we see that this was characterized by changes in locomotion during social preference, mean heart rate, and mean respiration rate that was associated with more segregation between association networks, and was significantly observed only in control males, meaning that stress is also disrupting this relationship. But overall, what all these findings means is that stress vulnerability is modulated by both duration of exposure and biological sex, and that manifests differently across different modalities. So initially, it was implicitly assumed that the way that the body responds to stress, either it's a dose-dependent response, a threshold-dependent response, it was never addressed whether this was a dynamical process that differed across different bodily systems. Then we can also see that our brain and behavior results show 2 complementary frameworks of stress vulnerability, either a dose-dependent response or just a threshold-dependent response that upon a stress exposure, things change. And lastly, we are providing this translational framework where we can link preclinical stress research with theoretical models that exist, that have been proposed before of stress vulnerability. And with that, I would like to thank Malar and Rose for all the guidance in this project and everyone from the Cobra Lab and the Bagel Lab.
So if I'm a stress researcher and I want to test out a new drug I want to test ketamine in the model of chronic variable stress in mice, and I want to use both males and females, what dose should I choose?
I mean, it depends if you want to assess how you can treat, for instance, behavioral susceptibility, which I would suggest go for different doses of stress in males and females, right? Okay. Like if you want to rescue those type of phenotypes. But if you're looking into other things, like the picture is not that clear of just saying, oh, use this dose of stress, meaning that it depends on what mechanism you want to target, I would suggest.
Obviously, I think a lot of like the initial characterization of the model, and correct me if I'm wrong, but it was probably based on behavior, right?
Yeah, yeah.
This model's what we think of as some like, if we anthropomorphize it, looks like human depression, right? So
That's what humans can assess.
As well as we can assess,
Mhm.
as well as whoever was initially developing these protocols. They're like, oh, it stopped struggling, it's probably depressed, right? So I don't know, do you think that like if you like how do your imaging findings kind of align with what we know from human imaging? Like, do you think that the same kind of thing is at play, that in humans with depression there are these different mechanisms that underlie the different phenotypes, whether you look at, like, obviously we diagnose depression based on feeling and behavior, I guess, but
yeah, well, I mean, so your question is like, what's a complex question?
sorry.
I mean, if we think about it as a translational perspective, I do think that this work kind of like confirms that in this model, we do need sex-specific doses of stress if we're just looking at behavioral phenotypes, right? Because they do look like they are emerging at different durations. So if we want to do this from a translational perspective and we want to treat these disorders, if you just want to remove like the behaviors, then I would suggest using dose-dependent, sex-dependent durations of stress.
Your right findings also speak to the recent expansion of functional networks paper.
Yeah,
Um,
yeah.
describe that.
Yeah. So like there was this previous article describing, so this was deep phenotyping 6 humans with depression. And what we see in there is that there's enlargement of the salience network where it starts to like encroach the DMN, which is similar to what we see here. One of the things that they were not addressed in that paper was this like a sex-specific finding. They never told like which sex these people had. But overall, what we see is that it seems to be a a specific thing that in this case, we observe it in males, where we see that the spatial correlation between the DMN and the limbic network and the salience network is changing upon stress exposure. So it's kind of also parallelizing things that we see in humans.
Two questions. You mentioned about depression at the beginning and what your test actually is anxiety-like behavior. Have you considered using real like depression-like behavior to test this animal and do correlation work? And yeah.
Yeah, I mean, in this case, we only consider like the open field and the social preference test, mainly because it was an expansion of a previous project. But yeah, we can definitely test this with other metrics of depressive-like behavior, like sucrose preference, more social interaction measures to see how these mice are actually socially active. Are they withdrawing from other mice in more like complex environments? Yeah.
This is you measure 3 factors reflect these. So which factor is the best reflect the anxiety-like behavior?
That's a good question. So I think both, the first two, because what we're seeing with the first one is a behavior that is being done more in the borders. And what we're seeing with the second one is a behavior that is being less done in the center. So both factors are pointing to the same direction, that mice are avoiding the center, and they're also doing less behaviors, like obviously in that place, but they're also, when they are in the borders, they do more of those behaviors.
So like simple correlation study looking at these factors, these 3 factors, compared to the center time, like conventional measure, and to see which is the best correlation.
I guess with the mean center PLS results that I showed, so this is looking at just patterns of covariance across all these behaviors. And what we see is that, for instance, in females, anxiety-like behavior is more associated with head scanning behaviors, whereas for males, it's associated with crouch and scanning behaviors.
So you think this 3 factors measure is better than conventional measure, just looking at whether they enter or the center time, or using that to measure anxiety-like behavior?
Yeah, but I think like
Is better than that.
I think both things are like giving us like good information about these mice. They're complementary. One downside is that, for instance, if we just look at time spent in the center, we see that males and females are affected similarly by stress. Whereas if we look at these more nuanced behaviors, this is where we start to see these sex differences emerge. So while I do think that these measures are really insightful into like just finding out whether this mouse is more anxious than another one, I do think that these types of behaviors help us get us a better understanding of what is happening in a sex-specific way in these mice.
So all of these behaviors are scored during the like at a specific time, right? During those tests.
What do you mean scored? So we record them? No, we don't have it. Like we wish we had that. That would be great. But yeah, so one of the greatest limitation of this study is that these mice are in a really controlled environment. So they are stressed with their cage mates, maybe adding some more that they don't know of. But all the time, they are never interacting with other mice. We didn't record them like more naturalistic environment, like just direct social interaction in their cages or something like that. This is just like we got them for our behavioral tests, and then they got back to their home cage.
Well, is it automated scoring? Or said land and it's like a team just like saying, yes, this, correct. It's like automatic. It can identify what behavior it is, like.
Yeah. So we record the behaviors, and then what we do is that we track the different body parts, and then we use just like AI, we can call it, to define which all of these behaviors like a sub-second scale. And it just gives you like the mouse is doing this.
HRV.
It was measured using a pulse oximeter during scanning. So during the functional scan, we use a pulse oximeter. Yeah, it is a possibility because we use a mixture between isoflurane and dexmedetomidine, and both things influence the cardiovascular system. We try to keep it as similar as possible across mice, but it might be related also like the way that they respond to the anesthetics.
Assuming some your do functional scans prior to your CBS.
So the main reason that we didn't do it is that we still have not, we don't have a clear picture of how to analyze functional data in rodents, mainly because of the anesthetic issue. Like we don't know like the reproducibility from one scan to the other one in order to do it at both time points. So just for practical issues, we decided to do it at the post-stress time point, and that was mainly like the reason, yeah.
So you use 3 ways to stress the mouse. So I mean, like, yeah, there's like the pulse shock, shocking, and then the tail, and the other thing. Do you think like each of them has a different way of stressing mice? And then like, do you think within your cohort, could there be some type of like how one mouse could be more vulnerable to this type of stress persistent?
That's a really good question. We didn't think about that before. Like, we do like each stressor brings something different to the picture. So for instance, in the foot shocks, they are stressed with their cage mates and other stress bodies that they don't know, they've never met before. So it brings like a different picture than just being hanged by the tail for 1 hour. So yeah, each stressor is bringing like a different thing to the big picture. But yeah, it could be a possibility that one of them is more susceptible to a specific type of stressor. Yeah. It would be definitely interesting, but I think for that we would need like a bigger sample to test subtypes in there. I don't know who wants to stress more mice. Yeah.
Looking at HRV, so at 28 days, the female mice showed greater HRV rates compared to the male mice, but 15 days the male mice were more than the female mice. So I'm wondering, long term, do you think that is something that would remain at that threshold, or do you think like female mice would probably show increased stress and male mice would show diminishing like increased stress? Does that make sense?
So you're saying like, are these things like dynamic adaptations?
Does it level off? Or do you think like female mice like stress increasing at a longer term with female?
Yeah, no, I do think it's like a dynamic thing. So there's like a trending, almost like difference in females that they have increased variability in heart rate, meaning that they might have like a protective mechanism probably against stress. But then it succumbs at 28 days. But I do think that they are dynamic. I don't think it's like a dose dependent thing, but at least in females, it seems to be like it's really dynamic upon like the different durations of stress. Yeah.
This is what discussion point rather than question. I was wondering in terms of when you think about sex differences, you're saying that there's a higher vulnerability and females push it down. Then if you think about it in terms of sex hormones and sex chromosomes, so you sort of start off for this like your presentation saying that females, women are at a higher vulnerability compared to males who having depression. When you think about like later on in life and for menopause, like there is this increase in this increased risk of depression in women. To what extent, but also here, these are young mice that are also sites for females, so they're still cycling.
Yeah.
To what extent do you think it's sort of like there's a role of sex hormones and then sex chromosomes or something else?
I think it's a mixture of both things. So there is data showing that, for instance, testosterone is kind of like protective for males at 6 days of exposure, for instance. So if you remove testosterone, like they become susceptible to stress. So I do think it's a mixture between both things. So there are also some studies showing like these, I don't remember the mouse model that plays with the chromosomes and the hormones, the four genotype. Those show that it's like this complex interaction between hormones and chromosomes, and it's not only hormones that determine stress vulnerability, but it's a mixture between both things that are happening all across neurodevelopment. So we see like there are regions that are larger, for instance, just depending on chromosomes, and these might be vulnerable to stress, but there are also regions that are just influenced by hormones that are also vulnerable to stress. So it's a really complex picture. That's what I think.
Well, Liz, that was a great talk.
Thank
very
you.
much. A lot of great getting together. Thanks a lot.
Thank you.